Hello everyone and welcome to AP Biology with Mr. Jennings. Today we're going to talk about macromolecules. Now this is a topic most people cover in their general biology class, but you really don't approach it from a AP perspective. And really this video here is just to serve as a brief review. We'll talk a little bit about what it means in AP biology, but in reality we're mainly going to talk about these topics when we come across them. So we're not going to really get too in-depth. We're going to get a little bit more so than what you do in your general class. Alright, so we're going to break this video up into 4 sections: carbs, lipids, proteins, and nucleic acids. So you're going to see 4 different videos on this topic, and just keep an eye out for those. So first off, let's just make sure we understand what the heck a macromolecule is. Macromolecules are just simply large molecules. The prefix macro means large. Pretty self-explanatory. These are some of the largest molecules in chemistry, and they're put together in a very special way, which we'll discuss. They tend to be very complex, but not always. I'll say this. It's not always complex. Example, DNA. DNA is a very basic molecule, comparatively speaking, but they can be very complex. So let's talk about that as we get to it. So all macromolecules have a uniform type of structure, at least a nomenclature we talk about. Except for lipids, all macromolecules have what we call monomers, which are one subunit. So there's a variety of them out there, we'll talk about them. And all of these, since they are monomers, have polymers, which are a chain of repeating monomers. We use a lot of the terms in polymers with plastics, but in reality, we do it in organic just as well, in organic biology. So going between monomer and polymer requires of a unique process known as dehydration synthesis. This is the removing of water, is what it sounds like. It's to build something by removing water. So this is a unique way that all organic molecules are built. And this is how it works. So this here is an example of a dehydration synthesis of what we're going to learn as carbohydrates. So if we look here, at the end of this carbohydrate, we have a hydrogen, and it's bonded to an oxygen. And we have an oxygen bonded to a hydrogen. Well, under enzymatic pressures, these 3 things are going to come together to form water. So basically, you're going to pull water out of the most available location. And to get into the mechanics of it, it's a little complicated. But for the basic thing is, if you see an OH on one molecule and an H on the other, that's likely going to be the location of the dehydration synthesis. And a bond is going to take place. So the water is going to come out, and this carbon will then bond to this oxygen, making one really large carbohydrate. And that's the dehydration synthesis in a nutshell. Now this can work in reverse. So dehydration synthesis is when you take away water. Hydrolysis is when you add water back. And that is pretty much like how you can dissolve certain compounds with water, but when you add water back under certain conditions, it chemically breaks it apart, causes this thing to split back up and form unique compounds yet again. So let's talk about the first major classification of macromolecules. So carbohydrates. Carbohydrates is probably the one that you're most familiar with, or at least the one that has the most terminology. It's tend to be the target of most fad diets, like Atkins, keto, etc. South Beach, yeah. So carbohydrates are pretty much just your sugar and starches. There's a few other things, but for the most part, sugar and starches. The elements, well, they're carbon, hydrogen, oxygen, and typically in a monomer form we see 1 carbon for every 2 hydrogen for every 1 oxygen. That's typically the breakdown of carbohydrates in monomer form. Now it's obviously going to be distorted when it's in what we call a disaccharide, or in polymer form, because we're taking out hydrogens and oxygens. Now the function of carbohydrates, the primary one is short-term energy storage. This is our primary function. So if somebody asks what's the function of a carbohydrate, more than likely they're asking what its function is. It's going to be short-term storage of energy. Now there is certain groups that do structural support, and we'll talk about those in a moment. The monomer for carbohydrate is monosaccharide. It just means 1 sugar. That's all it translates to. Saccharide is an old-fashioned word for sugar. Something needs to be called saccharin, which means it's a little too sweet. You may hear about movies and plays. Like somebody said a movie is too saccharine, I mean it's too sweet. So monosaccharides and disaccharides. These again are your monomers and slightly more complex, but not as complex polymer. There's three major monomers of carbohydrates. The famous three are glucose, fructose, and galactose. And we pretty much talk about all these. Disaccharides are any of these two monosaccharides linked together via dehydration synthesis. So almost all of our forms of sugar are some form of fructose, galactose, or glucose and or galactose bonded together. And look here, these are all chemically speaking the same. So these are all largely isomers of each other, just different forms of isomers. So look here, glucose and galactose. And galactose, the hydroxide group, this thing right here, is on the top. And glucose, it's on the bottom. And fructose, the functional group, is right here, and instead of over there. So, and it's also a 5 carbon ring, and this was a 6. So they look chemically similar when you look at them in the formula, but when you look at them in structural, they look totally different. So that's how you know you got an isomer situation. All right, polysaccharides. Now these here are the massive ones. This is where all the complexity comes in. So polysaccharides are massive. They have energy storage functions. And those would be like your starches. The classic example of a starch in plants is amylose. Amylose is the #1 plant starch. It's also the reason we have what we call amylase. It's an enzyme that breaks down amylose in our mouths. And it is our primary form of starch, is our glycogen. Now, glycogen is our typical animal starch. Not really we talk about too much there, but it does matter a lot. Now, for support, there's a variety of them out there, but the primary one is cellulose. Cellulose is what's in plants. It's fiber. It's when you rip apart celery, you find little strands sticking out of it. Those strands are cellulose. It's what gives plants a structural support, and it's also the thing we cannot digest. So it's kind of very important to see. Alright, so this is a quick little wrap up here. So when we look at carbs and organic molecules in general, these macromolecules, carbs are probably the most familiar and kind of the most beat up of the macromolecules. They're very important though. We talk about how we develop carbs, that's photosynthesis and cellular respiration, how we develop and utilize them. They are a primary energy storage molecule. Without carbs, you don't function properly. Now, carbs can be inherently a little difficult with our body because our body processes them to storage, long-term storage in the form of fat. But in the whole, you need carbs to live, so you just can't cut them out altogether. Now, when we look at carbs, you make sure you understand the structure of the carbs matters and how those carbs are formed. So the difference, for instance, like between a starch, like amylose, and a fiber like cellulose is just a couple of different things. It's an isomer of each other. One different branching, and all of a sudden you've got cellulose. So structure really dictates the function of these molecules. Alright, so the next video you're going to see here in a little bit, hopefully, is going to be lipids. So you're going to see the lipid video next. That's going to be very similar to carbs and a little different at the same time. Keep watching. Thank you.